So we talked about DNA. Now we're going to talk about cells. We're going to talk about being able to manipulate the epigenetics, your epigenetics, using electromagnetic energy. And if you don't know what that is, please stay tuned. Twitter, Human Internet 1, Parlor, Human Internet, PATRIOT'S.com. This is the show tab for big tech. You can find the archive notes here under Read, Watch, Think for Yourself. PATRIOT'S.com. You will have a central point to go to should they continue to censor this channel. That being said, let's get going. Today we are going to talk about systems and methods for electromagnetic radiation for influencing cellular structures. And we are going to talk about cellular reactions and gene expression using varying oscillation frequencies. We are going to revisit the DNA resonance that we discussed on the last episode. We're going to go through a little bit about DNA and RNA construction. We are going to talk about protein 3D structure formations because they're all made from the same stuff and it's just a difference of how they're folded. And the reason that applies is because when you start looking at how electromagnetic energy influences the hydrogen bonds, then we begin to see how potentially they're able to, on demand, steer, create, and or destroy how proteins are folded out of the same 20 amino acids. And if that blows your mind, good. If you don't understand what I'm saying, please stay tuned because this is important. We're going to talk a little bit about multi-photon ionization. And the reason we're going to talk about that is because right now regulation restricts the transmission of electromagnetic radiation as it relates to allowable limits being underneath what they consider ionizing radiation. So everything we understand for wireless communication functions under the non-ionizing category. And the ionizing radiation is restricted for medical applications. And the multi-photon ionization basically means that they can use non-ionizing radiation and through the pulsation of it, transmit enough energy using non-ionizing radiation to actually ionize components in biology. And that has everything to do with how they're able to manipulate the hydrogen that are the bonds that are required to create the folds in the proteins of DNA, or the folds of DNA that make proteins, is the way that that's said. We're going to talk about epigenetics. We're going to talk about phenotypes, which has to do with genetic expression. We're going to talk about soliton waves. We're going to talk a little bit about plasma formation. So if we can get through all this in 2 hours, then we're going to be doing all right. So first things first, let's get going. We're going to look at this patent right here. And this patent is System and a Method for Electromagnetic Radiation for Influencing Cellular Structures. Now, this is a medical environment, medical setting. You have a radiator. It can be microwave, millimeter wave, gamma wave, X-ray. It can be light. It can be infrared light. It can be ultraviolet light. The electromagnetic spectrum covers all of those things. And we will look at the electromagnetic spectrum briefly. So let's look at what this patent has to say. So this patent here, System and a Method for Electromagnetic Radiation for Influencing Cellular Structures. A system and a method for the present invention requires the use of a generator in combination with a radiation unit. So this is just an antenna and something that generates energy to that antenna to radiate electromagnetic waveform energy onto a target tissue, for example, a cell structure. During the radiation of the target tissue, in accordance with the predetermined titration-like protocol, which we'll get into in a minute, the influence of the waveform energy on the cell structure is periodically monitored. So this is a real-time monitoring when they emit electromagnetic radiation onto your body, into your body, targeting cells or cell structures. They're able to monitor that. The protocol is stopped when the cellular structure has been transformed or morphed into a desired phenotype. So what is a phenotype? This is important to know. We have to know what a phenotype is. National Institute of Health, National Human Genome Research Institute. A phenotype is an individual's observable traits, such as their height, eye color, and blood type. The genetic contribution to the phenotype is called a genotype. Some traits are largely determined by the genotype, while other traits are largely determined by environmental factors. That's what we're focusing on tonight. The electromagnetic radiation being an environmental factor that's able to manipulate your cells and your genetic expression. A phenotype simply refers to an observable trait. Pheno simply means observe. It comes from the same root as the word phenomenon. And so it's an observable type of an organism and can refer to anything from a common trait, such as a height or hair color, to presence or absence of a disease. Frequent phenotypes are related and used the term, I'll restate this, frequently, phenotypes are related and used. The term is used to relate to the difference in the DNA sequence among individuals with the difference in trait, be it in height, hair color, or disease, or what have you. But it's important to remember that phenotypes are equally, or even sometimes more greatly influenced by environmental effects than genetic effects. So a phenotype can be directly related to a genotype, your genetics, but not necessarily. There's usually not a one-to-one correlation between the genotype and the phenotype. So the expression of the genetic code isn't a one-to-one match to you having a certain genetic code. Environmental factors come into play when what genetic code is activated versus what genetic code is not activated. And that is epigenetics, and we will look at that in a minute. So the epigenetic switches that tell the body or the DNA which pieces to turn on and which pieces to turn off ultimately make the phenotypes. There's almost always environmental influences, such as what one eats, how much one exercises, how much one smokes, etc. All of these are environmental influences which will affect the phenotype as well. So we're talking about electromagnetic radiation being able to manipulate your genetic expression, resulting in a change in your phenotype, in a phenotypic representation, however that manifests. It can be disease, it can be cellular health, it can be anything. So the present invention pertains to systems and methods for causing transformational or morphological change in cellular structures. To transform or morph, mutate, change cellular structures using waveform energy, radiation. Radiation. More specifically, the present invention pertains to systems and methods which epigenetically influence cellular structures. Epigenetics. What's that? In biology, epigenetics is the study of heritable phenotype changes that do not involve alterations to the DNA. Epigenetics do not alter the DNA, right? The Greek prefix epi, over, outside of, or around, in epigenetics implies on top of or in addition to the traditional genetic basis for inheritance. Epigenetics most often involve changes that affect gene activity and gene expression. Very, very important. Gene expression is the process by which information from a gene is used in the synthesis or the functional gene product. These products are often proteins, but in non-protein coding genes such as transfer RNA or small nuclear RNA genes, the product is, let's read this because this kind of ties in with that vaccine, don't it? Doesn't it? Gene expression is summarized in the central dogma first formulated by Francis Crick, blah, blah, blah. So basically, gene expression is used in the synthesis of a functional gene product. These products are often proteins. Your body's made of all of these proteins, but they also make things like enzymes and everything else. But in non-protein coding genes, such as transfer RNA or small nuclear RNA, the product is a functional RNA. So we're going to look at RNA and DNA and see why all of this is important. Let's move on because we don't have time to get into the depths of genetic expression. So it says the present invention is particularly, but not exclusively, useful for systems that use electromagnetic waves to alter the resultant functionality of in vivo in the body or in vitro out of the body target tissues. So we're gonna breeze through this, and we're gonna jump around and look at some different things to try to define just exactly what this patent is saying is possible with focused electromagnetic waves. It's well known that electromagnetic and sonic waves can have many different effects on tissue for many different reasons. Moreover, in some cases, consequences of radiating tissue may be permanent. Apart from applications where sonic waves may be used to induce or suggest changes in a particular mood or state of mind, we're talking about sound waves at this point, being introduced into a biological in order to change the mood or the state of mind. It is also recognized that both sonic and electromagnetic waves can be employed to cause transformative or morphological changes in cellular structure, the cells in your body. Not surprisingly, many of these changes may be very beneficial. Thus, within the medical community, there is increasing interest insofar as the extent to which such changes may be employed to beneficially alter the functionality of cellular structures. With the above in mind, consider the specifics of sonic waves and the effects they can have on tissue cells, cellular structure. From a physical perspective, a tissue cell can be likened to a mechanical system. The importance of these physical observations is that sound, acoustic pressure, acts to exert a force against a tissue, the mechanical system. And this will happen whenever a sonic wave is incident on the tissue, applied to the tissue. Thus, the tissue will be influenced by the externally applied force that are associated with the sound wave. As a mechanical system, each individual cellular structure, the tissue of the cell, has a natural frequency at which it will oscillate if it is not subjected to continuous or repeated external force. This will be the case unless the cellular structure is somehow otherwise subsequently stimulated, such as when a sustained vibratory frequency is applied to the cell. Most noticeably, there will be no such diminishment in the vibratory frequency when it's at or near the natural frequency of the cell structure. Instead, in this case, a resonance condition is established wherein the vibratory nature of the cell responds and is sustained and possibly even amplified. So we're talking about using sound waves or electromagnetic waves, finding the unique frequencies, the right modulation techniques to resonate your cells, to excite them and manipulate the expression of those cells. Here's an interesting piece. From a biological perspective, each cell, in this case, example, a liver cell, will have observable characteristics which naturally result from the cell's environment. A set of these observable characteristics is generally referred to as a phenotype. So now they're talking about unique characteristics of a cell that is within a particular organ. That's how specific phenotypic classification can be, a cell in a particular organ. Further, it's known that a set of characteristics for a defined phenotype of a cell can be epigenetically influenced by externally applied forces. Moreover, this can happen regardless whether the cellular structure is influenced in the body or outside the body. So, it's well known, it's also known, electromagnetic radiation of all frequencies will have a physical effect on cell structures, albeit sometimes negligible. Of particular interest here, however, are the physical effects that result in an identifiable, transformative, or morphological change in a cellular structure. We're talking about electromagnetic radiation now. For this purpose, high-frequency radiations are of unique interest. High-frequency radiations, insofar as their penetration of the human body is concerned. In light of the above, it is an object of the present invention to provide a system and a method for using radiation to epigenetically influence tissue cells to thereby alter the functionality of an in the body or out of the body target tissue. It is another object of the present invention to provide a system and a method for using a radiation waveform energy source to influence a change in a target tissue by tuning an electromagnetic wave to a wavelength that is related, similar to the natural frequency of the target tissue. We're talking about tissue resonance, cellular resonance, organ resonance. It is still another object of the present invention to provide a system and a method for radiating waveform energy in accordance with a predetermined titration-like protocol. So that's just, we radiate you, we take a sample, we see what we did. We radiate you again, we take a sample, we see what it did. We radiate you again, we take a sample, we see what it did. To epigenetically influence tissue cells for the transformation or morphology, mutation, of the target tissue into a desired phenotype. So changing the cell expression into an expression or a result that is desired. Being able to control how your cells replicate and or express themselves by activating and deactivating certain components of the DNA within that cell. That's what this is saying. Another object of the present invention is to provide a system and a method for using radiated waveform energy to epigenetically influence tissue cells, which is easy to use and commercially cost effective. So it says here, it's well known that waveform energy, radiation, right? Electromagnetic waves create forces. They exert pressures on an object when the radiation is applied to the object. It's also well known that these external forces can cause changes to tissue structure. This invention is related or based upon this phenomenon. It says here, for the intent of this invention, this present invention, target tissues of interest may be in the body or outside the body, maybe a cellular structure of the human body. It may be an individual cell, or it may be a group of cells within the intracellular tissue matrix that supports the cell. So what does that mean? Well, you have cells and then you have support structure cells that ultimately combine together to make things like an organ or veins or nerves or anything else. And that's what they're talking about. There's segmenting these things. It is an actual organ cell, for an example, or is a support structure cell within that organ. They're making that distinction because they can identify them uniquely based on the resonance characteristics. As envisioned for the present invention, target tissues may also be identifiable structures inside a cell, such as a chromosome. So now they're talking about being able to identify targets of interest like a chromosome, which is a bunch of DNA wrapped up into chromosomes. In such case, it is important to appreciate that as a mechanical structure, the cellular structure of a target tissue will have a unique natural frequency. So we're talking about using electromagnetic waves to resonate in a unique way, being able to target cells independently, exclusively from other cells, based on those cells or those structures' unique resonance. It says here, an objective of a protocol for this invention may be the creation of a type of stem cell. Being able to manipulate stem cells, to have the stem cells create certain types of cells. For example, they say a liver cell from an otherwise undefined or undifferentiated cell. In this case, the desired phenotype, the outcome, will be defined to have the requisite characteristics of a particular type stem cell that is desired. For example, a liver cell. So what does that mean? That means if they know the unique resonant characteristics of a liver cell, they can go to a stem cell and resonate that stem cell using electromagnetic radiation. They can resonate it at the liver cell resonance frequency and make that cell, that stem cell, become a liver cell. And that ties back into the brain Q patents that we were looking at, where they were literally talking about being able to control the growth and the placement of brain cells using magnetic fields. So this is another example of where they're defining in patents how this is actually possible with electromagnetic energy. As another example, protocol may be to terminate the viability of a cell structure, meaning to kill cells, such as by killing cancer cells. Now you can put 2 and 2 together, folks. If they can kill cancer cells, but they can grow liver cells, you think they can cause cancer? Just a thought. Just a thought. Other examples can be cited. In each instance, however, and regardless of the specific outcome that is desired, the present invention employs waveform energy radiation, electromagnetic radiation, for the purpose of epigenetically influencing target tissues for its transformation or morphological change, mutation, into a structure that corresponds or matches the desired phenotype. So they talk about being able to use wavelengths up to 10 wavelengths 10 to negative 25 meters up to 10 to the third meters. So everything basically in the traditional electromagnetic spectrum. 10 to the third meters is radio waves, low frequency radio waves, and 10 to the negative 25, you're up in the gamma and X-ray zones. So everything in between. And we'll look at electromagnetic radiation spectrum here in a minute. It says that the radiation may be either continuous or pulsed, and the tone of the radiation may either be a pure single frequency or a complex multi-frequency. Now, what does that mean? A pure single frequency isn't going to give you a great amount of bandwidth if you're going to look at a wireless communication system, for example. A complex or multi-frequency transmission will. And when we look at orthogonal frequency division multiplexing, OFDM, for those of you that want to look that up now, you can search OFDM and you will see that they're able to generate complex waveforms that are able to send large amounts of data from point A to point B. And if the receiver device understands what the OFDM timing and other unique characteristics are, then it literally gets the ones and zeros, and you get things like being able to watch YouTube. But hey, it's just an example. Let's keep going. It says here that a system for using radiation waveform energy to influence cell structures. What's needed for that? These things include components for generating and directing radiation to the target tissue. Well, we looked at beamforming the other day. We're going to look at beamforming again as time goes on. We looked at 3D beamforming. And as time goes on, we're going to look at how beams are steered around and directed. We're going to look at how beams overlap and create unique interference waveform patterns. So directing radiation can be done now with modern communication systems. Components for monitoring the target tissue. Well, we've gone over the Google patents. We'll go over them again as it relates to their solely gesture recognition, for example, using things like ultra wideband and millimeter wave frequencies to be able to sense temperature at multiple points around the arm, being able to sense micro twitches of muscle, being able to sense heartbeat, being able to sense blood flow, being able to sense all kinds of interesting things down to a millimeter or sub-millimeter scale. Components for monitoring target tissue, and a computer for controlling the generator and the radiation unit. So that could be anything in the cloud. It could be your cell phone. It could be Wi-Fi. Google has patents to actually overtake other transmitters, other devices, other smart connected devices. Google can take over their transmission protocols and use them as radar fields, as well as allow them to continue to communicate. Google has those patents as well. And we'll look at those as time goes on. We've looked at them already, and that's outside of the scope of what I'm sharing with you today. So it says here that the radiation, right? They use the example of lasers, but that's only one spectrum, one piece of spectrum in the electromagnetic spectrum. They say that typically a distance will be greater than 10 millimeters, which means it's no contact at distance. Greater than 10 millimeters. It could be a mile away. for example. If it was in space, it could be a lot further than that. Hashtag satellites, 5G in space. A method in accordance with the present invention begins to identify target tissue to be influenced, including the cell structures, and by defining a desired phenotype for the target tissue. A natural frequency for the phenotype can then be determined and referenced in the literature. So what does this mean? It basically means that they can look and identify a target tissue, they can measure that target tissue, and then they can induce a change in that tissue and then measure it again to ensure that they're getting the right expression of that tissue. That's what they're talking about. That's really the meat and potatoes, so to speak, of this patent. It says that the radiation can be pulsed. Well, if you look at how wireless communication works today, all of it's pulsed. Pulsed electromagnetic radiation is basically modern wireless communications in a nutshell. And the subsets of that are how it's modulated, but it's all pulsed. Even continuous wave is pulsed when you look or consider the variations in amplitude, frequency, or polarity. You can view those as pulses. But pulse in and of itself is its own form of modulation. They have pulse modulation as well. And we'll look at those things as we keep going down the path of 5G. We're going to spend August looking at wireless energy. And what does it mean? What does it look like? We are going to get deep into the weeds as it relates to wireless electromagnetic radiation, because this is what we're constructing. This is what our economy is going to be based on. So, hey, guess we should know what we're dealing with, good, bad, or indifferent. You be the judge. So it says that the pulse, right, will have a predetermined time and a predetermined time interval, right? So a time that it's on and a time that it's going to be off. So it can be on for a certain amount of time and then off for a certain amount of time before it's turned back on again. And that's where you get things that look kind of like square waves. It's one of the ways you get a representation of something like a square wave. So the electromagnetic spectrum, I need to drop share because I don't think I checked the box. So I did check the box, outstanding. All right, so we're gonna watch this video from NASA posted on NASA's site. We're gonna watch a little bit about the electromagnetic spectrum. Credit to NASA.
Something surrounds you, bombards you, some of which you can't see, touch, or even feel. Every day, everywhere you go. It is odorless and tasteless, yet you use it and depend on it every hour of every day. Without it, the world you know could not exist. What is it? Electromagnetic radiation. These waves spread across a spectrum from very short gamma rays to X-rays, ultraviolet rays, visible light waves, even longer infrared waves, microwaves, to radio waves which can measure longer than a mountain range. This spectrum is the foundation of the information age and of our modern world.
So here's just a real basic diagram of the electromagnetic spectrum. This is what we're talking about. What we're talking about is this invisible essence, if you will, being able to manipulate your biology in many, 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 many different ways.
Your radio, remote control, text message, television, microwave oven, even a doctor's X-ray, all depend on waves within the electromagnetic spectrum. Electromagnetic waves, or EM waves, are similar to ocean waves in that both are energy waves. They transmit energy. EM waves are produced by the vibration of charged particles and have electrical and magnetic properties. But unlike ocean waves that require water, EM waves travel through the vacuum of space at the constant speed of light.
So kind of important. These things travel really, 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 really fast. They travel at the speed of light in the vacuum. They slow down very, 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 very gently. They'll slow down a very little bit when they're in an atmosphere because they'll hit particles and whatnot, water, dirt, dust, all of that stuff. So they'll slow down and they'll get diffracted a little bit, but they can make accommodations for that. They can sense that upon reception, send that back to the transmitter, and then make the necessary accommodations.
EM waves have crests and troughs like ocean waves. The distance between crests is the wavelength. While some EM wavelengths are very long and are measured in meters, many are tiny and are measured in billionths of a meter, nanometers. The number of these crests that pass a given point within 1 second is described as the frequency of the wave. One wave, or cycle, per second is called a hertz. Long EM waves, such as radio waves, have the lowest frequency and carry less energy. Adding energy increases the frequency of the wave and makes the wavelength shorter. Gamma rays are the shortest, highest energy waves in the spectrum. So, as you sit watching TV, not only are there visible light waves from the TV striking your eyes, but also radio waves transmitting from a nearby station, and microwaves carrying cell phone calls and text messages, and waves from your neighbor's Wi-Fi, and GPS units in the cars driving by. There is a chaos of waves from all across the spectrum passing.
Hashtag surveillance. So we've got all of these things that are ongoing all the time that nobody sees, nobody really thinks about, nobody really gives a second thought to. Wi-Fi from the neighbor, microwaves from your cell phone or your own Wi-Fi, light coming out of your TV and or from your ceilings or your lamps, GPS, AM, FM radio, digital television broadcasts. All of these things are forms of focused man-made energy that, if modulated in the right ways, can influence your biology. And the only regulations at this point are how strong are the electric fields, how strong are the magnetic fields, and what is the frequency spectrum that's being used, because anything that's over what's considered ionizing radiation can't be used in a public environment. It must be used in a medical space. And I can show you, what that is. It's right here. So ionizing radiation starts at ultraviolet, right here, 12.4 electron volts. I believe is where ionization, where they make the break. And we can search this real quick just to be sure. Ionization, ionizing. Ionizing versus non-ionizing radiation. So let's see if Wikipedia will tell us. Yeah. Radiations with particle or photon energies less than 10 electron volts are considered to be non-ionizing. So non-ionizing, right? Right up to ultraviolet. Anything underneath that is legal. AM, FM radio, Wi-Fi, microwaves, radio waves, infrared waves. Anything above that, it's controlled. Tanning booths are kind of, they have special requirements, and then you got medical x-ray. Anyway, so that's the difference between ionizing and non-ionizing. That's really the only regulations: electric fields, magnetic fields, and whether or not it ionizes and how much it heats up tissue. Those are the only regulations at this point. And we've looked at lots of patents in the last few shows that discuss very, 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 very small amounts of energy in these magnetic fields or electric fields that are able to manipulate cells and biology. So let's continue. It's enough on the electromagnetic spectrum for now. We'll go back and revisit that video as time goes on. So shorter wavelength radiations are more energy intensive. We just learned about that, right? You've got, here's your let's blow this up into its own screen so we can see this. So here's your frequencies. It's not gonna let me. How great is that? So your frequencies are here, wavelengths are here, energy is here. We are looking at radio waves, right? We know all about radio waves, VLF, LF, high frequency waves, very high frequency waves. Right in here is where we are dealing with like cell phones, Wi-Fi, radios, televisions, things like that. And when we get up here to EHF, that's where we're getting into the millimeter wave stuff. Super high frequency and extremely high frequency. This is where millimeter waves, you get 60 gigahertz for your Wi-Fi, 60 gigahertz for these radar chips that are coming out. Some of them are in the 40 gigahertz, some of them are in the 70 gigahertz. These are in the EHF frequency band. And this is where we are moving into. This is what's publicly coming out into the commercial space. And these are the powers, right? This is the energy per photon that's going to be transmitted in a very focused kind of a way. And right around here, ultraviolet, near ultraviolet light, this is where the breakout is, ionizing versus non-ionizing radiation. And so it's going to be completely legal based on current FCC standards. It's going to be completely legal to be able to put out these higher energy, more focused wavelengths out into the public space, as long as they meet the SAR requirements, specific absorption rate requirements. And the electromagnetic field densities are ridiculous. They're well above what's necessary to manipulate biology. We'll go into that in more detail over the next few weeks when we start fusing in nervous system manipulation with this electromagnetic discussion that we're going to be having over the next approximately 8 shows. So it talks about fluence here, and fluence is just established as a term, right? That's used to define photons in a unit area. So what they're saying is that they're able to control and regulate the amount of photons that are able to be applied to tissue, regardless of the frequency spectrum that they're using. The number of photons is different than the energy that's associated per photon. What do I mean by that? Every one of these values, right? This is a value for one photon. One photon is one wavelength. So in 1 second, I will get more photons if I'm transmitting at 300 terahertz versus 30 terahertz, because I have more waves passing in 1 second. So I will get more photons. Same thing if I've got down here in 30 gigahertz versus 3 gigahertz, more in a range that I think we're all familiar with. And there's more energy. So there's a lot more going on, in my view, that the FCC needs to take into account than what's currently regulated, because we are dealing with very, very sophisticated directed energy systems that are now providing modern communication infrastructure that do things like mess with your genetics, can determine whether or not a gene turns on or turns off, can tell whether, or can shape and control the production of certain cell types in the body and then steer them around, as well as being able to tap into, manipulate, and modulate the nervous system to do things like animate arms and legs, all of which we've covered in the last 3 or 4 shows. So that's the world we're living in now, folks. So it says here to identify and establish an efficacious and fundamental frequency for target tissue, it needs to be determined, right? So importantly, a fundamental frequency will be characteristic of a particular phenotype of a desired cell. So that means that if you have a cell or a cell structure, if it's expressing itself one way versus another, if it's healthy versus unhealthy, it's going to have a different frequency. It's going to have a different fundamental frequency. So what they're really saying is if they want a particular type of cell to work better, then they can measure the frequency it's resonating at originally, right? And then they can apply electromagnetic energy to get it to artificially resonate in the way that they want it to resonate to make it work better until it does it all by itself. Conversely, they can do the opposite. They can take healthy cells and make them sick. Same kind of a thing. It talks here about an octave evaluation, right? And that's what we looked at last episode, was being able to use any electromagnetic spectrum that a transmitter is capable of transmitting, and then by using an exponent of 2, right? Dividing the resonant frequency by 2 or multiplying it by 2, you are able to get the frequency shift down into the band with which the transmitter is able to transmit. So for Wi-Fi, it'd be 2.4, 5 gigahertz, 60 gigahertz. For cell phone, it could be 900 megahertz all the way up to 6 gigahertz. And for modern Wi-Fi, now we're dealing with 60 gigahertz. And when we're dealing with cell phones have to be able to scale between all of those things, because they'll be able to talk to all of those things. So a cell phone really is more dynamic than your Wi-Fi, or more dynamic than your cell tower, because it can talk to a 60 gigahertz router. It can talk to a 5 gigahertz router. It can talk to a 2.4 gigahertz router. It can talk to cell sites. And the phones are typically built universally now, where they're digitally provisioned. So they come off the line being able to tune into any commercially available cell tower. And as we move into the world of 5G, things are going to be scalable within much wider frequency bands. And that's something called software-defined radio. And so it's going to be very easy to find an octave, a derivative of 2, if you will, an exponent of 2, up or down, from the resonant frequency of a cell or an organ or DNA, and get exactly what we're looking for as it relates to being able to resonate biological systems in the body. So it says here in this patent, they talk about sensors, right? Being able to measure this stuff as they're making these manipulations. And they talk about titration-like methodology. So what is a titration-like methodology? And this is something that you'll see in a lab. Right? Volumetric analysis. It's a common laboratory method of quantitative chemical analysis to determine the concentration of an identified analyte or a substance to be analyzed. A reagent, termed as a titrant or a titrator, is prepared as a standard solution of a known concentration and volume. This titrant reacts with the solution to determine the analyte's concentration. So how is this applied to what we're talking about right now? What this looks like is they would be able to draw blood, take a biopsy, saliva sample, swab your nose, and they would be able to analyze that material using a titration methodology to see what's going on with your genetics, to see what's going on with your DNA, right? That's one example. Another one is polymerase chain reaction. And for those of you that don't know what this is, a polymerase chain reaction is kind of the same thing. This is a widely used method to rapidly make millions to billions of copies of specific DNA, allowing scientists to take a very small sample of DNA and amplify it to a large enough amount to study it in detail. Now, where do you think this is being used today? Anybody? Makes you wonder why we are all being tested. The new coronavirus tests. And if they can get this down to 5 minutes, guess how many companies are going to require this? At some point, if we don't say no, you may have to get one of these tests in line while you wait to go into a store to buy food. Right? It says here that tests take hours to run, so they need to have better ways to run tests, right? Once the rapid form of these PCR and antibody tests are created, results could be ready in under an hour. There are rapid PCR tests, but they are not quite available yet for the new coronavirus. And this is an older article because we've seen now that they have test validations 15 minutes, 10 minutes, 5 minutes. Once these are deployed, it will become an expectation to be tested. Now, if you have a population who is being tested on a regular basis and you have systems in place commercially that are able to do things like ionize structures in the body through multiphoton ionization, or be able to resonate cells or DNA to get phenotypic expressions, turning on and turning off genes, it'd be very easy to do these kinds of things. Have your population take regular tests, and you begin to study exactly what these electromagnetic systems can do to biology. And the laboratory then becomes open air, open society laboratories. Just my opinion. Asterisk, asterisk, punctuation point, fine print, fine print. But when you look at the nature of what's going on and the push for what it is that is 5G and this whole push for electromedicine, this is what I see, folks. I see social control that requires mandatory testing so that they can do things like collect your DNA through polymerase chain reaction techniques and or collect your DNA or genetic information to perform titration-like methodologies, right? So that they can understand what's going on with your epigenetics while you bathe in focused, targeted electromagnetic energy, which according to this patent and some of the others that we've read over the last few episodes are able to do things like resonate your DNA, identify genes and identify certain proteins and protein structures. Now we're seeing that they can turn on and turn off epigenetic switches, activating certain DNA so that they express and turning off DNA so that they don't express. Being able to do things like take stem cells and convert them into organ cells. In this case of the patent, they said it was a liver cell. Being able to do things like destroy cancer, just reverse those. What if they wanted to weaken your liver or weaken your pancreas and grow cancer in your body? If they had the ability to test on a regular basis, they would know exactly what works with exactly what demographic. Black people versus Asian people versus Mexican people. People that eat this type of diet versus people that eat another type of diet. People that drink this water from this location versus water from another location, city to city. When you think about what's put into municipal water supplies and how that could potentially be used to manipulate epigenetics. And then you tie in the ability to accelerate or stunt epigenetic expression using electromagnetic energy that's being forced upon us at breakneck speed while you're putting in all of these other raw materials, quote unquote, into the food supply, into the air, and into the water to act as building blocks. Oh, we're going to get to building blocks here in a minute. But before we get to building blocks, we need to look at the other method that they're able to use to understand what's going on with your epigenetics, and that's bioelectric impedance. Bioelectric impedance, commonly used method for estimating body composition, in particular, body fat and muscle mass. Body impedance analysis is a weak electric current flows through the body, and the voltage is measured in order to calculate impedance or resistance of the body. Most body water is stored in muscle. Therefore, if a person is more muscular, there is a higher chance the person will also have more body water, and it leads to a lower impedance. Since the advent of the first commercially available device in the mid-1980s, this method has become popular, owing its ease of use and portability of equipment. Now, why is this important, folks? Why is this important? I'm going to clean up tabs here real quick, so bear with me. Fitbit, bioelectric impedance analysis. Here's a search term. It's in the docs, should you choose to look at it. Fitbit, here's a help link to Fitbit. Body fat percentage is the mass of your fat divided by your total body mass. It's estimated by using a bioelectric impedance analysis, which sends a small safe signal through your body. Generally, a body fat percentage is between 10 and 22% for men and 20 to 32% for women. How accurate is Fitbit's bioelectric impedance analysis? This is in a watch, folks. It doesn't stop with measuring the water content in your body fat. They're looking at the electrical characteristics of what's going on in your body, and the difference is how good is the software. So that's Fitbit. Here's Samsung's watch, their Galaxy watch. Patently Apple does an article. Samsung invents a body composition measuring device that may be coming to a future version of the Galaxy watch. A body composition measuring device, which is sort of a healthcare device, measures body composition using bioelectrical impedance analysis that obtains a ratio between quantities of fat and moisture by using a flow of low energy alternating current waves based on an electric resistance difference between adipose tissues and non-adipose tissues. What else? Oh, it's the article for polymerase chain reaction test for coronavirus. We just looked at that. So going back to this paragraph here, there are different ways that sensors can be used to evaluate what's going on with your body. And in particular, as it relates to the epigenetic components that basically control and regulate phenotypic expression, how your cells express themselves in the body. So it doesn't stop at the watches. It obviously will apply to headphones and or body that can do the same type of sensory functions. You can go look at Charles Lieber's patents. It's out of the scope of the discussion today, but go look at them. That's what they do. They're cellular sensors that do things like understand changes in pH, that do things like understand chemical signaling that goes on inside a cell, which has everything to do with how your genetics, your DNA is functioning, protein formation, where the proteins go, what chemicals are sent to trigger what piece of DNA needs to be accessed in order to generate the right protein or the right enzyme or the right whatever. Because all of that is run from the software that is your DNA. Speaking of DNA, maybe we should digress here for a second and look at DNA. We have an hour left, so let's look at DNA. Why all this push on DNA the last few episodes? And we'll get there. So DNA, nucleic acid, right? They're biological molecules essential for known forms of life, right? DNA and RNA. They're present in the nucleus of all cells. They control all the chemical changes that take place in cells, DNA. The kind of cells which is formed, such as muscle, blood, nerve, etc., that's all controlled by DNA. The DNA controls what is created by the cells in your body. The kind of organism which is produced, buttercup, giraffe, herring, human, etc., is controlled by the DNA. It's a very large molecule made up of long chains of subunits. They're called nucleotides. Each nucleotide is made up of sugar called deoxyribose and a phosphate group, PO4, and an organic base. This is important. We're going to get back to the phosphate group in a minute. So here's what a basic building block looks like. These are the organic bases: adenine, thymine, cytosine, guanine. Everything out of DNA is made out of A, T, C, and G. Adenine always forms with thymine, and cytosine always bonds with guanine. Always. Talks about some subtle variations. Sugars, ribose and deoxyribose. Ribose only has 5 carbon atoms. Deoxyribose is almost the same, but lacks 1 oxygen atom. We're going to keep going because a lot of this is outside of the scope. The functions of DNA. It's the genetic material. Therefore, it's responsible for carrying all of the hereditary information. This is the software repository for your body, the DNA. It has the property of replication, essential for passing genetic information from one cell to its daughter cells or from one generation to the next. Crossing over produces recombination. Changes in the sequence and the number of nucleotides causes mutation, which is responsible for all variations in formation of new species. It controls all the metabolic reactions of cells through RNA and RNAs directed synthesis of proteins. A molecule of DNA is formed by millions of nucleotides joined together in a long chain. Now, these are the phosphate groups. Remember this. These are going to be the helixes, right? These are the backbones. So you've got the ladders in the rung of a ladder of DNA, and you've got the ropes on the side, and they twist and make a helix. Well, the phosphate groups are part of that rope structure that connects the bases together. DNA usually consists of double-stranded nucleotides. Sugar phosphate chains are on the outside, and the strands are held together by chemical bonds between the bases. So we're looking at just the general construction flattened out of DNA. Notice the phosphate groups. This is going to be important in a minute. Different types of bonding, pairing up, the role of phosphate linkage. Nucleotides of both DNA and RNA are covalently linked. That means they have 2 bonds. Covalent bonds are 2 bonds that linked through a phosphate group. They're bridges in which the 5-phosphate group of one nucleotide unit is joined to the 3-hydroxyl group. Important, hydroxyl, that means hydrogen. We're going to get into why hydrogen is so important here in a minute as well. There is a method to the madness. Of the next nucleotide, creating a phosphodiester linkage. So what does this mean? This means that the phosphate groups on the outside interact with the hydroxyl groups, which consist of hydrogen. And the reason why this is important, folks, is because that's how proteins are made. Based on how these things bond with each other, determine what protein is made from the same 20 amino acids. The same 20 amino acids make up all proteins. The difference in the proteins, and there are hundreds and hundreds and hundreds of them, the difference is how they fold. And the way they fold has everything to do with the way these phosphate groups bond to hydrogen. And that is important because hitting the hydrogen with electromagnetic waves breaks the bonds, which means you can then start steering and shaping what proteins look like. And we're talking about DNA creating the building blocks in your body. And this is on a layman's scientific level, how they're able to steer and direct and guide biological functions in your body, up to and including generating things like a liver cell on demand from your stem cell repository. Double helix, sugar phosphates on the outside, the bases on the inside. This looks like the DNA that we are familiar with. We'll keep going. DNA structures. They're able to resonate these things based on all the way down to a gene. So DNA structure, you could have strands and strands and strands of this, and then you have certain genes that are within the strands. They can resonate down to a specific gene. They can resonate to a particular. And we are almost through. mRNA, let's stop here because mRNA is part of these new COVID vaccines, right? Messenger RNA, mRNA, carries information about a protein sequence to ribosomes, the protein synthesis factories in the cell. So these mRNA vaccines, right? They're going to go in and they're going to be uptaken by the ribosomes, and they are going to tell the body, tell the cell what proteins to make. And the way the vaccine works is they're going to enter synthetic code via mRNA, one example, there are others out there for these vaccines, to go into your cells, tell your cells to make a protein that is like the coronavirus, so they say, could be anything. We're talking about genetic code here, as well as being able to manipulate that code with electromagnetic energy. So who knows what the hell is going to happen once it's been put into the body. It's like the ultimate Trojan horse, bait and switch. Hashtag Project Warp Speed. Give me a freaking break, man. The synthesis of mRNA takes place inside the nucleus, and after its synthesis, it's exported out where it attaches to ribosomes for protein synthesis. So the mRNA goes inside the nucleus of a cell, and after it's worked on, it's recognized and processed, unzipped, rezipped, repackaged, with the right components put onto it, it travels to the ribosome and then it makes the protein. And that's really what mRNA does. And if you remember all of the things we looked at with the Visa Institute, this should look familiar to you. Transfer RNA. Transfer RNA is some interesting stuff. We'll go back maybe one day and look at this. So we will continue on. Why is this important? Protein 3D structure. The big picture about DNA. Keep in mind, we're looking at ways in which you don't necessarily have to modify DNA, but you can control how cells are expressed, how proteins are made, what proteins are made without necessarily modifying DNA using electromagnetic energy. And we'll continue. To understand the basic principles of protein, three-dimensional structure, and the potential of their use in various applications in academia or industry, we first need to look at the picture by defining the 4 levels of a protein structure. The different structural levels are mutually dependent on each other, together creating an extremely complex network of interactions between hundreds and thousands of protein atoms, solvent molecules, and often various ligands and metal atoms. Interesting. Now we're talking about potential use of nanotech, right? Because they use ligands to encapsulate nanotech, and nanotech can be metal, it can be polymers, it can be any number of things, it can be genetic material. But if we're talking about protein structures that use metal to generate the actual structures, now we're talking about being able to potentially infuse nanotech into proteins because they are that small, where we're building complex circuits into the body, literally into the proteins that make up the cells in your body. Yeah, I see you out there in chat. Let them say what they're going to say. Chances are they haven't been listening very long. So I encourage you to listen. Keep coming back, because over time you will hopefully see what I see. What I see is a threat to our species, period. So we'll continue. The 20 most common amino acids found in proteins are joined together into a polypeptide chain during the process of protein synthesis. 20 common amino acids. Most proteins are made from these 20 common amino acids. So what they're saying here is these are essentially the formation of the peptide bonds catalyzed in the ribosome. So blah, blah, blah. We don't really need to know about that. Secondary structure elements, right? Define tertiary structures. So now we're kind of getting into basically what this means is they're starting to describe how a protein, if it's just a strand of DNA, once the DNA is made, it's replicated, it's a long rope-like structure, then it's going to fold into a unique structure, into a unique formation that makes a specific type of protein. There are currently known three-dimensional protein structures that have been classified into around 1,300 different unique folds. So they've classified the proteins, right, into about 1,300 different unique folds. Yeah, I know, guys, this is a lot of information, and a lot of it's scientific-y, and it's a lot. We're going to go over it again, but you're drinking from a fire hose at this point, and you should. And we'll go over it again, and it'll get easier and easier and easier and easier. Just stick with me on this. This stuff is very important. We're not in Kansas anymore, folks, and we all just need to realize that and accept what we're faced with at this point. So 1,300 different unique folds of proteins, and they've all been classified. And on this website, they talk about a protein database, protein structure databases, a short overview, bioinformatics, resources on the internet. These are commonly available. Growth of unique folds per year as defined by this group, right? So it's a long time ago, they started looking at proteins in the 70s. And as computation and modeling and testing and evaluation and synthetic biology has all sort of taken off in the last 2 decades, boom, now we're up at 1,300 classified, categorized proteins. So we looked at we haven't looked at amino acids yet. 20 amino acids and their role in proteins. Each of the 20 most common amino acids has specific chemical characteristics. They're based on the propensity of the side chain, blah, blah, blah. They list them all here. And just for posterity, here they are. And some of these things you'll see like in ingredients in food. Glutamine, tyrosine, tryptophan, phenylalanine, I think, leucine, glycine. Some of these sound familiar. Okay. Hydrogen bonds. Why hydrogen bonds? We're going to get there. It says here, for a hydrogen bond to be formed, 2 electronegative atoms, for example, in the case of an alpha helix amid the amide N and the carbonyl O, right, have to interact with the same hydrogen. So they're talking about creating hydrogen bonds. And it says water molecules may also be involved in the stabilization of protein structures by making hydrogen bonds with the main chain. Talk about salt bridges. And we'll continue. We're not going to get into too much more because this stuff is, I mean, people go to school for this stuff for years to figure this crap out. And you're literally getting a download of what I looked at for 8 hours today before the show. I got on my computer and was like, what are we going to talk about? And boom, this popped up in my face. So this is what you're getting. So it says here that, let's see if I can find hydrogen on here. Yeah. It says basically the point of all of this is to show you this. There are groups within a DNA construct or a protein construct that form hydrogen bonds. And one of the main forces, hydrogen bonds are one of the main forces in the stabilization of the secondary structure in proteins. The hydrogen bonds are shown on the right figure as dashed lines. So what we see here are the hydrogen bonds. And what does this look like, right? So you've got DNA that's made in a double helix, and then it folds over, right? And these hydrogen bonds are what bend and shape these things into different proteins. So helical structures include the helix, and it's stabilized by hydrogen bonds. It's really what it's saying here. So it says here that these polypeptide chains, they're kept together by a network of hydrogen bonds between adjacent strands. The stabilizing hydrogen bonds between adjacent strands are shown as dotted lines. So when you think of DNA as just being a ladder that's twisted around, right? Well, if that stuff makes proteins, which actually makes all of the things that make you what you are, skin, liver, brain, heart, lungs, blood, all of those things are different proteins. They're made from DNA. They're made from the same general amino acids, but they're folded, they're constructed uniquely. And that's what makes them different. A blood cell versus a heart cell versus a lung cell, on and on and on and on and on. And those structures, there's a key component of why they're structured the way they are, and it has to do with hydrogen and hydrogen bonding. And hydrogen bonding is just an ionic bond. It's not a covalent bond. It's only linked with one. It doesn't have a parallel link. And it makes it very sensitive and very delicate. So the role of hydrogen in DNA, why is this important? Because we're talking about being able to manipulate the epigenetics, being able to tell the cell what DNA to use and then how that cell expresses it. It doesn't change the DNA, it just manipulates the usage of it and the ultimate construction of the result, meaning the proteins. Hydrogen bonding. Importance of hydrogen bonding. It's important in many chemical processes. Hydrogen bonds hold complementary strands of DNA together, and they are responsible for determining the three-dimensional structure of folded proteins, including enzymes and antibodies. Right here, hydrogen bonding. Multi-photon ionization. Write this down. Look into this. Multi-photon ionization. Multi-photon ionization of atoms. It results. All right, I guess we'll click on it and read. Present overview, currently understanding of multi-photon ionization of atoms, begins with an introductory section, explain the background, blah, blah, blah. It says here that it's not going to tell me, and it's behind a paywall. I'm not going to be able to download this. So we'll go back to the abstract or the here. Simultaneous absorption of several photons constitutes a further generalization of photoionization. Photoionization is just basically using the light spectrum and the electromagnetic spectrum, something like a laser. It was discovered as a result of the conjunction of two new situations, blah, blah, blah. It gets better. We have microwave ionization of hydrogen atoms. The reason I showed you multi-photon ionization is because when we look at the electromagnetic spectrum, right? Let's go back and look at this electromagnetic spectrum. Tell you what.
They transmit energy. EM waves are produced by the vibration of charged particles and have electrical and magnetic properties.
Let's just look at a chart. The electromagnetic spectrum. And we know that around ultraviolet light, that's the separation between ionization and non-ionization, non-ionizing and ionizing radiation. Well, multi-photon ionization means that they can use non-ionizing radiation and pulse it in the right way so that cells, DNA, protein, whatever, absorb that energy, and ultimately it creates an ionization occurrence in a cell or in the body. And that's what this is saying. Microwave ionization of hydrogen atoms. So this is going to be able to unfold or direct the folding of those proteins that are made by the DNA, which is a way that they can manipulate the epigenetics, which ultimately has everything to do with how healthy you are. That's just one example of how healthy you are. It can change your mood. It can change your organ function. It can change the ability of your blood to carry oxygen. It can do all kinds of things when they can start manipulating the epigenetics. Microwave ionization of hydrogen atoms is a process of electron ionization of excited hydrogen atoms by an electromagnetic microwave field when tens or hundreds of photons are required to ionize one electron. Even if a microwave field is relatively weak, this multi-photon ionization is much more efficient than a direct one-photon ionization at high photon energy. So FCC, if you're listening, just because you've segmented and made legal non-ionizing radiation, this document basically says that it's easier to ionize hydrogen with multi-photon ionization than it is to use one photon, which is how they segment and distinguish between ionizing radiation and non-ionizing radiation. The energy of one photon to displace an electron. And this just said that multi-photon ionization is more efficient than a direct one photon ionization at high photon energies to ionize one electron. Such a rapid ionization happens due to diffusive growth of electron energy generated by dynamical chaos in the classical system. Quantum effects can suppress this diffusion with the emergence of photonic localization, which is similar to the Anderson localization in disordered solid state systems, blah, blah, blah. So what they're really getting at here is they actually say it, the quantum effects of photonic localization were first observed by the group of Koch in 1988. So what they're really getting at here is they're dancing around this idea of quantum-based medicine. Quantum energy, quantum-based energy systems that are able to do things like move electrons around. And that's what we're made of. Protons, neutrons, and electrons that make up the elements, that make up the basic building blocks for everything we understand as matter. And that's what they're talking about, being able to manipulate here with microwave radiation using multi-photonic ionization. That's what they're talking about here. So some other documents, microwave ionization of hydrogen atoms, the breakdown of classical dynamics for high frequencies, behind a paywall. Can't get to this. But here they talk about 36 gigahertz. They used an experiment, right? Being able to use microwave energy to ionize hydrogen. That has everything to do with how three-dimensional protein structures are folded, up to and including enzymes and antibodies. The hydrogen holds together DNA and is responsible for determining how DNA is structured in three-dimensional space that create proteins, including enzymes and antibodies. Highly excited atoms in the electromagnetic field, also behind a paywall. So let's go back to this. The system and a method for electromagnetic radiation for influencing cellular structures. And we've now gone through approximately an hour of supporting information to illustrate just maybe what they're talking about here. Directing electromagnetic radiation at distance to an organism to affect specific cells based on the cell's resonance, to epigenetically modify that cellular expression and change the phenotype And mechanically, what that looks like, in my opinion, is doing things like being able to manipulate how the hydrogen bonds are made or destroyed as it relates to DNA folding into different proteins that make up the human organism or a biological organism, up to and including antibodies and enzymes. Now, the mRNA, back to that. If we know that that mRNA is used to transport information from the nucleus of a cell or from the inside cell, right, to the ribosome, to then have the ribosome make the protein, and we're going to introduce foreign mRNA into the body. And we now know that there are technologies available whereby they can emit electromagnetic radiation to direct and steer how those proteins are going to ultimately be formed. And we are going to go in and reprogram cells to immunize people using DNA and RNA-based therapies. You tell me what you think is possible. And you tell me if you think that what's going to be injected as advertised is going to be the only thing that's possible. Hashtag warp speed. Alex Azar, lobbyist for Big Pharma, who knows all about electroceuticals and electromedicine and this mRNA crap. And that's just the vaccine. This isn't day to day, folks. Bathing in this stuff. Can you imagine how fast cancer could be formed with something like this? Conversely, can you imagine the cure for cancer just being voila? Your home router, your phone, whatever senses that you've got growth of cancer cells and it automatically emits the resonating frequencies to kill it while you're sitting there with your family? Watching TV or while you're asleep, you would never get cancer because it would be able to identify it by sending a series of resonant frequencies through your body and then reading the reflection of those transmissions to understand what's resonating and what's not. And if the cancer resonance frequency went off, a red flag went up during the scan while you're potentially asleep, it just delivers the therapy for you and you don't have cancer. That's the world we could live in. But instead, we're talking about genetically manipulating the entire species for God knows what. And we are going to be judged for it, in my opinion. Let's talk about this one. A system and a method for titrating in vivo cellular reaction and gene expression using varying oscillation frequencies. Cellular reaction and gene expression, manipulating those things using varying oscillation frequencies. Well, let's just read what I've highlighted here because the rest of it really is similar to what we just went over in detail. It is well known that human beings will physically react to sonic energy. Bear in mind that this last patent referenced sonic energy and said that electromagnetic radiation basically does the same thing. So, hearing is but one example of this phenomenon. It often happens, however, that the human reaction to sonic energy is not immediately perceptible. Instead, the perception is the result of a longer-term evolutionary process. In this last category, in vivo cellular reaction and gene expressions have been attributed to the influence of sonic radiation. I'll read that again. In vivo, in the body, cellular reaction and gene expressions have been attributed to the influence of sonic radiation. Of particular interest here are cellular reactions that result in a phenotypic differentiation of target tissues. Sonic radiation being able to change the phenotypic expression of target tissues, just like the electromagnetic radiation. Tesla, energy, frequency, vibration. Energy, frequency, vibration. It's the same thing. Look up photoacoustic, look up thermoacoustic, look up electroacoustic. You're really going to find it under thermoacoustic. Voice to skull technology is the use of microwaves to expand and contract tissue, to heat it up and allow it to cool down rapidly, to generate sound waves in the body. The Air Force uses this for covert communications. It's called voice to skull technology. The medical field is using thermoacoustic and photoacoustic imaging techniques because they have higher resolution than just using ultrasound alone. They will use light or microwaves, pulse them into the body, and use a sonogram tool, what you would see on a pregnant mother's belly, right? An ultrasound transducer to measure what the microwave energy is doing, or the light energy, the photonics, what they're doing in the body. Because when they transition through the body, they generate longitudinal waves, they generate physical waves, they generate sound waves. And so they use an ultrasonic transducer to pick that stuff up, and it provides a high definition image. The medical field is using this stuff. You can look it up. Microwave acoustic imaging, thermoacoustic imaging, photoacoustic imaging, microwave hearing. That would be the Air Force patents. I can show you those. And the reason I'm saying this to you is because we're looking at something that says that the influence of sonic radiation is able to do things like manipulate the phenotypic expression of target tissues up to and including cells. And there are technologies that prove that they work. Introducing microwaves into tissue and then reading it with an ultrasound transducer in the medical field provides unequivocal proof that microwaves are in fact generating sound waves in the body, and therefore, if modulated correctly, could do things like manipulate the way your cells express proteins, enzymes, antibodies, and everything else. Hashtag 5G. Apart from the phenotypic cell differentiation that can result from exposure to sonic radiation, sound waves, it's also known that meditative states and or alpha brainwave relaxation states are greatly influenced by sound waves. Really? So in addition to phenotypic cell differentiation, how your cells express themselves, it can also induce changes in your cognitive state, your ability to relax. Could it stress you out? Could it make you irritated? Could it make you go to sleep? Could it keep you awake? Could it make you feel euphoric? Could it give you senses of intense pain? Could it induce fear? Sound waves, which microwaves can induce in the body, and they use it in the medical field today. You can look it up. Maybe we will. We have time. There's only one more highlighted paragraph to go. On this point, consider the effect music can have on an individual. Also consider the effect an exposure to a loud, abrasive, and or startling noise can have on an individual, regardless of whether the sound is instantaneous, repetitive, or continuous. I will add to this binaural beats, using sound waves from two different locations, offset by a certain number of hertz, and the brain picks up on the change. And they can do this with sound waves. We will look at that in detail when we start looking at autonomic nervous system manipulation. And we're going to get there also. Although a wide variety of protocols for sonic radiation, sound wave therapy, have been, it says have 5 been, that's interesting, can be proposed, it is It is axiomatic that for beneficial therapy, sound waves must be effectively directed onto the proper target tissue. So it has to be directed. So directed energy, beam forming, interfering waves, beam steering. 5G, same kind of technology, phased array antenna technology that's able to take a radiation beam and steer it and focus it to a very narrow range. With this objective in mind, an effective delivery of sound waves necessarily requires a controlled exposure to the radiation, sound wave radiation, right? This control in turn requires the employment of pre-planned operational parameters, frequency, intensity, and duration. Frequency, pretty cut and dry. Intensity, that's the power field. And duration, how long are we applying the radiation for? These cross apply to electromagnetic radiation. Preferably, this is all done with minimal requirements for patient supervision. Well, because we're moving into the age of automation, I don't think the patients are going to have much of a problem. They won't have to worry about it. So going back to NASA, before we look at photoacoustics and thermoacoustics for medical imaging, just to unequivocally show you that this is a viable technology that's in use today, electromagnetic field interactions observed with the human body. If you have not looked at this, you should. I would like to point your attention to the document number NASA CR 166661. This was published in April of 1981, and it has the summation of a select number of studies for the preceding 2 decades from the 60s and the 70s. And in this document, you will read all about the fun things that they were willing to disclose in this NASA report, physical and physiological foundations, adverse, benign, and curious effects of electromagnetic radiation on the human organism, beneficial effects. A lot of the beneficial effects in this document are redacted. They don't show them. In this document, they show ionization potentials of elements, and these are the voltage fields that are required to ionize these things. They're in Roman numerals 1, 2, 3, 4, 5, 6, 7, 8. And they've got all of the elements here. So when we look and think about multiphotonic ionization, they can just pick one. Here's some compounds for ionizing. Oh, look, there's water. Water. Ionization potential 12.56. 12.56 electron volts, basically, to ionize water, which is what we're made of, most of. So I want to show you pearl chain formation because this was a heavy hitter for me when I saw it the first time. It says here it's on page 81. So 81, 82. This gives the mathematical calculations for pearl chain formation. But I want to show you the diagram. So, man, this thing is lagging. The computer is not keeping up with the scroll. Here we go. So pearl chain formation, really, this is what it is. It's able to take the dipole, basically, of a molecule. Let's just call it water, for example. You have a positive side and a negative side, and they're random, right? It says without a field, they're random. Well, when you induce an electromagnetic field, you put them all in alignment, where all the positives are on one side and all the negatives are on another. You can actually control the dipole and the orientation of things like water molecules in the body using electromagnetic fields. And this was way back in the 60s, 70s, and 80s when they were working with this stuff. Here you go. Negatively charged particle together with a double positive layer form an electrically neutral hole, but an induced dipole, which arises from a charged particle, enters an electrical field, linking of the orientation dipoles in the direction of the electrical field. So what is this saying? Negative on the inside, positive on the outside. Negative gets pushed to one side. Obviously, you're going to have an equilibrium. And when it's induced into an electrical field, they line up and they all orient the same way in the direction of the electrical field. And this is basic as it relates to being able to spin up cellular functions in the body. So this is a 125-page document. It's in the show notes. I suggest you download it and look it over yourselves. I started a lot of my journey looking at this document. Electromagnetic field interactions observed, effects and theories. Electromagnetic field interactions with the human body. So let's look up thermoacoustic imaging. Thermoacoustic imaging. A strategy for studying the absorption properties of human tissue using virtually any kind of electromagnetic radiation. Alexander Graham Bell, you know, the telephone guy, first reported the physical principle upon which thermoacoustic imaging is based a century earlier. So this guy, Theodore Bowen, 1981, right, proposed it. But they're saying Alexander Graham Bell noticed it and reported it 100 years before, in the 1800s. He observed that the audible sound could be created by illuminating an intermittent beam of sunlight onto a rubber sheet. Shortly after Bowen's work, which was done in 81, was published, other researchers proposed a methodology for thermoacoustic imaging using microwaves. In 94, researchers used an infrared laser. Oh, an infrared laser. Hashtag Face ID. Apple, 30,000 lasers to measure your face. Infrared lasers to produce the first thermoacoustic images of near-infrared optical absorption in tissue. Makes you wonder what the brain-computer interfaces of tomorrow are going to be. If we're staring at our phones anyway, why not just pulse them with the infrared and do things like generate thermoacoustic effects in the face and the nerves and the skin and the brain? Because infrared passes through the skull and can go centimeters into the brain, meaning it can interface with the cortex. Pulsed microwaves produced the first fully three-dimensional thermoacoustic images of biological tissue. 1998, Indiana University Medical Center. The following year, they created the first fully three-dimensional thermoacoustic images of cancer in the human breast. Again, using pulsed microwaves. That's how cell phones work, folks. That's how Wi-Fi works, folks. That's how digital TV works, folks. That's how satellites work, folks. Pulsed microwaves, pulsed millimeter waves. So, the process by which thermoacoustic waves are generated. Biologic tissue is irradiated by an energy source. It's absorbed by the body. The source of energy is nonspecific, but typically consists of visible light, near infrared, radio waves. Oh, it's hypnotic. It's so hypnotic. Or microwaves. Dun, 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 dun. Biologic tissue is irradiated by an energy source that is absorbed by the body. The source of the energy is nonspecific, but typically consists of visible light, near-infrared light, radio waves or microwaves? The absorbed energy is converted to heat, which raises the temperature of the tissue, typically by less than 0.001 degrees Celsius. Well, that is very, very well within the FCC requirements. They're on the order of degrees of Celsius. You can't heat up tissue by degrees of Celsius, not 0.00 N degrees. So what, 3 orders of magnitude? Or we'll just say orders of magnitude within range, below the legal limit they're able to use for imaging when we're talking FCC regulation here. The increase in the temperature of the tissue causes the tissue to expand in volume, however, just slightly. This mechanical expansion produces an acoustic wave. Oh, sonic radiation, I think was the term they used in that patent, to do things like change your epigenetics and your phenotypic expression of cells, as well as change your cognitive state, how focused you are, relaxed you are, what brainwave state you're in, alpha, beta, theta, delta. Acoustic waves, sonic radiation, that propagate outward in all directions from the energy or from the site of the energy absorption at a velocity of sound, at the velocity of sound in biologic tissue. So they're able to generate ultrasound in the body by using light, infrared, radio waves, microwaves, millimeter waves. We might as well just add those too. They can create ultrasound in the body by heating up the tissue less than 0.001 degrees Celsius and allowing it to cool, and then heating it up again, and then allowing it to cool, and then heating it up again and allowing it to cool. And you get this pulsed radiation. On, off, on, off, on, off. And you do that at a high enough frequency. I'll explain it. It's right here. When the tissue is irradiated with a pulse, the acoustic frequencies that characterize the acoustic wave span a range from 0, which is 1 pulse width. Example, a 1 microsecond pulse produces acoustic frequencies from 0 to approximately 1 megahertz. That's just an example. Which means that if they pulse microwave radiation at 1 millisecond intervals, based on the time that they wait to pulse it again, they're saying that they get between 0 and 1 megahertz ultrasound. That's ultrasound, right? 1 megahertz is way over 20 kilohertz. Anything over 20 kilohertz is considered ultrasound. So they can pulse in microseconds and generate the entire audible frequency, sound waves in your body, including infrasound, which is below 20 hertz or 30 hertz. Infrasound is 1 to approximately 30 hertz. And from 30 hertz up to approximately 20 kilohertz is audible sound. That's the range that humans hear in. Anything above 20 kilohertz is considered ultrasound. And 20.1 kilohertz, 20.001 kilohertz, up to 1 megahertz is ultrasound. Shorter pulses produce a wider range of acoustic frequencies. Frequencies greater than 1 megahertz are referred to as ultrasonic. Now, that's probably specific to the medical field, but when you're dealing with audio frequencies, 20 kilohertz is the upper end of audible hearing, and anything over 20 kilohertz is ultrasound. They're saying here that frequencies greater than 1 megahertz are referred to as ultrasonic and are also associated with medical ultrasound applications. We will get into ultrasound in detail when we start looking at things like being able to provide artificial sensory perception, see, touch, taste, hear, smell. See, touch, taste, hear, and smell artificially by pulsing ultrasound into the brain. Artificial reality. with all five senses is possible by stimulating the brain with ultrasound, folks. And we will get there. Thermoacoustic imaging devices require a source of electromagnetic radiation, be it a laser or a microwave antenna, to deliver energy to the anatomy being studied, and one or more acoustic detectors coupled acoustically to the outside surface of the anatomy. The typical acoustic detector is an ultrasound transducer, which is commonly made of piezoelectric material that converts detected pressure to an electrical signal. Thermoacoustic waves are induced within the anatomy wherever absorption takes place, and the strength of these thermoacoustic waves is proportional to the energy absorbed within the tissue. Some of these waves propagate through the anatomy over a time interval, which is known as the time of flight, before being detected by one or more acoustic transducers. The exact time of flight is proportional to the distance between the absorption site and the transducer, assuming for a moment that each transducer is a point detector, right? So for any given time of flight, each transducer will receive a sum of the thermoacoustic waves. And this is how they're able to understand three-dimensional depth. They measure the time of flight. So they irradiate tissue, an organ, a baby, a whatever, and then they measure the sound waves that are generated by the tissue being heated up rapidly and cooled rapidly. And then they measure the amount of time it takes for those sound waves that are made to be received by the ultrasound transducer. And based on the time of flight, they're able to say what tissue was absorbed where, and then recreate a three-dimensional object. Thermoacoustic image of a human breast using microwaves to visualize normal glandular breast tissue, high resolution, high definition. So this is a proven, medically applicable phenomenon. That, according to this patent, is able to manipulate cellular reactions, phenotypic expressions, genetic expressions using various oscillation frequencies. In this case, they talked about sound in this patent. Let's see if they... Present invention particularly, but not exclusively, is useful for incorporation into systems that are used for electromagnetic or sonic radiation therapy. So the imaging technologies can also be used to provide therapy. And we looked at this patent that specifically discusses electromagnetic radiation for influencing cellular structures up to and including genetic expression, phenotypic expression, epigenetic manipulation. And we looked a little bit at how that might work as it relates to being able to manipulate how the hydrogen bonds are made or destroyed that ultimately change and or dictate how the DNA folds into proteins that make things like enzymes and antibodies, which are just two examples. As well as in this patent, it discussed being able to resonate a stem cell in a certain way to do things like create a liver cell from a stem cell and differentiate that stem cell as a liver cell versus something else, like a heart cell, a lung cell, a blood cell, or whatever. And this patent also discusses being able to destroy cells, and they use the example of cancer by influencing the epigenetic characteristics of the cellular expression.